I'm so happy to welcome everyone to the 2022 SeaTech Student Research Symposium. My name is Ann Simonis, and I'm really glad to be able to be here in sunny San Diego on the Scripps campus. We have a bunch of people joining us online as well, so if you're joining us from Alaska or Canada or throughout the United States, SeaTech's friends and family, welcome. This is awesome. I first came to Scripps over 12 years ago as a summer intern myself. And I remember the first year that I came to Scripps, I came to this very auditorium, Sumner Auditorium, and I saw one of the early SeaTech research symposiums. I remember the students from Mount Edgecombe came and they talked about bearded seals and bowhead whales, and I learned about all sorts of sounds I had never heard before. And they told me about how the animals' presence was related to sea ice coverage, and they shared obviously really impressive research. And I knew right then that I wanted to be a part of SeaTech. It was a couple years until I actually joined SeaTech officially. Dr. Josh Jones, who's in the audience today, invited me to the SeaTech family. And I can say that, I'm getting nervous, that SeaTech is really the best part of my professional life. I love working with the students. We have 7 students joining us today from Mount Edgecombe High School. We also have 1 student who's going to be joining us remotely from Oakland. The SeaTech family is growing. It's so nice to have expanded this program. We have a team of amazing mentors, graduate students from Scripps who've joined the team, really made this a fun and joyful experience. So I'm not going to talk about SeaTech anymore, but if anyone here or in the audience online is interested in creating similar experiences to what we've done here, please get in touch. I could talk about SeaTech for days. I'd be happy to have a coffee or talk about how we might be able to do similar work connecting students and researchers in other places, because really it brings so much joy to everyone who's involved. Okay, so I'm Ann Simonis. I'm going to step aside for now. I want to hand over the mic to Mr. Mahoney, their teacher. Oh, hold on. I have one thing to say. I have a couple things to say. Because we have limited time, we're only going to take one question after each student's presentation so that we can get through everyone. And I also want to acknowledge that I'm leaving this front door open. It might be a little bit noisy because there's some fans going on out there, but it's helpful for our lighting and also for air circulation. So I acknowledge that door's open, but we're going to keep it open. Okay, so that's it. Okay, Mr. Mahoney, I'll pass the mic.
Thank you.
Yeah.
Well, thank you all very much for coming. I'm Michael Mahoney. I've been a science teacher at Mount Edgecombe High School in Sitka, Alaska for 25 years. Mount Edgecombe is a very special place. We are a state-funded public boarding school of about 430 students in non-COVID years. And we're about 90% Alaska Native students, and they hail from about 120 different communities around the state of Alaska. And so they come and live with us for 9.5 months every year, and live in dorms and come to Mount Edgecombe to get a great education. With that said, living in a dorm situation, many of you know, can be a challenge. And so it is a status symbol for the students of Mount Edgecombe to let you know how many years that they have been there. So if you hear a senior say, I'm a 4-year senior, it does not mean that they failed their senior year 4 times. It means that they lived in the dorms, they stayed at the school for 4 years. And so that's a pretty big deal for them. I started this SeaTech adventure with Josh Jones back in 2008, and we've been doing this for 14 years now. And every year it seems to just get better and better. The support that we've received from the Whale Acoustics Lab at Scripps here and through NOAA has just been fantastic in helping us in this adventure of trying to get students to be passionate about ocean science. And so even though we've been doing a lot of acoustics, primarily, we're open to doing all kinds of adventures in science. And so with that said, let's get to the students. And so none of this could be possible without the support from Professor Bob Rex and his love for these students at Mount Edgecombe. And we just would like to say thank you very much for your support. All right. So we'll bring them up. Let's get started.
So I'm Haley Moses. I am a second year senior at Mount Edgecombe High School, and I'm from Fairbanks, Alaska, as you can see on the map. And yeah.
Hi, I'm Mary Simeon. I'm a third year junior from Bethel, Alaska, which is on the map up there. There's a picture of me working at the hatchery this year. That's me on a raft trip. That's a picture of me with a 20-pound dolly, and that's a picture of me and my mom.
Hi, I'm Jordan David. I'm a third year senior from Hooslia, Alaska. Shown in that picture right there. I am originally from the village of Hooslia, Alaska, which has a population of around 320 people. And I became interested in SeaTech when my junior year, Mr. Mahoney and my counselor kind of convinced me to do it. And I realized that I was already interested in marine science in the first place. So I thought, why not? And then here I am, my second year. And yeah, I plan to become a scientific researcher when I'm an adult and get my undergrad degree in biology and my graduate degree in possibly marine biology. And these pictures right here show me analyzing shellfish under a microscope. That's me and my roommate right here. And this one shows me in my traditional garb. And this is an aerial view of Hooslia.
Hello, I'm Rio Baca, and I'm originally from Port Orange, Florida, which is the picture in the background, although I'm currently living in Sitka, Alaska, which is located in the top right. And I joined SeaTech because I never really had the opportunity to learn about marine mammals or marine life in general, and I wanted to. And now I'm really glad I got to join and work with my mentor and learn about sperm whales and the growing threats of sound pollution and climate change.
Hello, my name is Shania Tanapi. I'm from Nanwalik, Alaska, and I'm a second year junior at Mount Edgecombe. I got interested in SeaTech because I told my counselor about wanting to be a marine biologist when I go to college, and she said SeaTech would be a nice class to take to help me in really choosing what I want to do.
Hello, my name is Keisha Askok. I'm a second year junior at Mount Edgecombe High School. I am from New Haven, Iliamna, Alaska, and that is shown right there in the top right corner. Bottom right corner is an aerial photo of New Haven. The middle top photo is of me on our lake, swimming in our lake. Bottom right photo is of me and my sister. And the top left photo, right photo is of my dog, and the bottom right photo is us on the lake. Bottom boat. Okay.
Yakoyala, Itian the Tiana Capella. Hi, my name is Tiana Capelli, and happiness to all of you. I am a four-year senior from Fairbanks, Alaska, but my family comes from Kayana, Alaska, and Likia Atoll Marshall Islands. I was the baby in SeaTech sophomore year in a class full of seniors and juniors, and I couldn't take the class this year, but I had the opportunity to finally come down and do the presentation in person. Here is a picture of where Kayana is,
And here's the midnight sun. Me and my cousins were kayaking at 1 in the morning in Kotzebue. And here's my dog, Shido, when we went blueberry picking, and a picture of me helping with cutting up a moose at school. I will be passing the microphone to Alex.
Hi, you guys. Here, let me share my screen super fast. Okay. Hello, everybody. My name is
Alex, we can't hear
Oh.
you. Hold on.
Hello. Can you hear me now?
How do I put the microphone on?
Hello. Can you guys hear me?
Yeah,
Yeah, we can hear you, Alex.
it's very cool.
Thanks. It must just be on the in person then.
I think it's my sound.
Can you hear me?
Yeah, they can hear. It sounds like on the online they can hear her.
Yeah, I think online they can hear, but not through the speaker.
Maybe it's the microphone on the podium needs to be present.
Oh, this one?
I don't know how to do that. Yeah, it worked fine yesterday.
Oh no.
We have to fix the output on HDMI.
No, not needed.
We do also have the.
Mic is up. Oh, okay.
It was plugged in yesterday.
There it's
Oh,
going.
but we were plugged in through this. Stand by.
Okay. This volume is up.
So let's select the projector as VGA. All right, there we go.
Okay.
There's my volleyball team.
Settings. Display. Noise. It should be. Yeah.
Can you guys hear me?
Do we have? Where?
Okay, let's go back to the.
Can this? Is this?
It's not on right now.
No. You could put that up against the speaker.
There. I think we could be there.
Okay.
Can you guys hear me now?
Oh, that's better. Yeah, here, turn the volume up a little more.
Okay, does that work?
Yes, we can hear you. Yes.
Okay. Perfect. Okay. Ever the COVID technological issues. But today I am presenting virtually because I was not able to attend the SeaTech trip because I currently have COVID, which really sucks. But I got introduced to SeaTech by Ann Simonis, and I will be presenting live from Oakland, California, which is where I was born and raised. So I have grown up in Oakland and am a junior at Oakland Technical High School. I am in the Engineering Academy, which you can see in the bottom left photo of me holding a vintage drill. I am also on the ice hockey team, which is shown in the photo right next to it. I sing in a choir, which is also shown in the photo next to that one. And I have always loved nature and wanted to protect it. The center photo is me with some friends at a climate march we organized. And as I look to the future, I think I want to major in environmental studies or sciences with a minor in economics. So thank you so much, and I will jump into my presentation. So hello, everybody. Today I will be presenting about Pacific white-sided and Rizzo's dolphin acoustic monitoring in a warming California current, which is a collaboration of NOAA Fisheries and Oakland Technical High School, my place of education. So my initial research objective was to classify Rizzo's and Pacific white-sided dolphin acoustic events and look for interesting patterns in the 2018 Passive Acoustic Survey of deep diving Adonisites. But first, a background on dolphin acoustic behavior. So odontocetes, toothed whales, dolphins, or porpoises used acoustic signals to hunt, travel, and communicate. These echolocations detect size, shape, and speed hundreds of yards away under the ocean. Some dolphins have acoustic signals that are unique and can be used to identify different species from acoustic data. The first of these species I will be looking at today is Rizzo's dolphin, or Grampus griseus. The second of these is Pacific white-sided dolphin, or Lagonarhynchus obliquidans. These two dolphins are found in the North Pacific Ocean. I studied these specific dolphins because they can be classified using their unique click signals. These graphics show the exact values of Pacific white-sided and Rizzo's acoustic signals. On the left, you can see a spectrogram with click number on the x-axis and frequency on the y-axis, and spectra on the right with frequency on the x-axis and amplitude on the y-axis. These are used to differentiate between odontocete clicks in data and identify unique peak and notch frequencies shown by the red arrows. The reason Pacific white-sided has two figures is because it has two different click types, whereas Rizzo's only has one. For data collection, I used drifting acoustic spar buoy recorders, or DASPers, but in this presentation, I will be referring to them as drifting buoys. The mast, flag, and buoy at the top and the subsurface buoyancy keep the drifting buoy upright and floating. The nylon line ensures that the recorders are reaching the depths of deep diving odontocetes. The upper and lower hydrophones are the two listening devices that capture the sound acoustic data. The data logger intakes the data collected by the two hydrophones. I used data from 22 drifting buoys deployed in the 2018 California Current Ecosystem Survey, or CCES, whose mission was to survey deep diving odontocetes. For my research, I repurposed this data to look at Pacific and white-sided and Rizzo's dolphins. This survey was done by the Southwest Fisheries Science Center, and CCES was conducted from the 26th of June to the 4th of December in 2018. These drifting buoys were deployed to predetermined locations along the western coast of the Pacific Ocean. The black square represents where each drift was deployed, and the black triangle represents where the drift's last known location was, or where it was retrieved. The black lines show drifts that were recovered, and the gray lines show the drifts that were sadly lost. In this presentation, I will be looking at drifts 7, 8, and 14, highlighted in red, which I had the time to analyze, and I thought represented the dolphin's distribution best along the California Current. For classification, I used PAMGuard, a software infrastructure for acoustic detection, which allowed me to flag dolphin clicks as either Rizzo's, Pacific white-sided, or unidentified dolphins. Here's the interface of a click that is clearly Pacific white-sided. The bearing time display with x-axis time and y-axis the angle at which the sound is coming in, the waveform display, and the Wigner plot, I am able to examine the clarity of an individual click and ensure that it is a dolphin. But the most interesting part, I think, is the spectra. This allows me to identify a species by its unique peak values. A spectra has frequency on the x-axis and amplitude on the y-axis. The solid lines in the spectra are from the hydrophones from a single click, and the shaded regions are the average of all the clicks in one event. Pacific white-sided and Rizzo's dolphin have unique peak and notch values, which mean by examining the three mean peaks of this example, with values at 22, 27, and 39 kilohertz, I am able to confidently label this as Pacific white-sided because of Melissa Soldavia's 2008 paper. After classifying each event using PAMGuard, this is the plot of Rizzo's dolphins found in the California Current Ecosystem Survey in drifts 7, 8, and 14. Red dots represent acoustic events detected. These classifications were generally found northern skewed, with none of them on the southern buoy. These are the Pacific white-sided dolphins found in 7, 8, and 14. From examining these plots, I noticed a northern concentration, which led me to compare this data to prior years to see if the location of Pacific white-sided dolphins had changed. It's not letting me change the slide right now. Hold on. There we go. Okay, thank you. To evaluate whether the location of Pacific white-sided and rhesus dolphins was consistent over time, I compared acoustic data from drift 7, 8, and 14 and visual observations from the entire survey, with red circles representing the acoustic events and triangles representing the visual events, to historic visual data from the NOAA stock report between 1995 to 2005 shown in gray. We can see that the data between 1995 and 2005 Pacific white-sided dolphins were spread along the entire US West Coast, but in 2018, Pacific white-sided dolphins were detected only north of Point Conception, marked by the yellow line, except for two outliers in the 2018 visual data. From the plot shown above, we can see that there has been a surprising absence in the southern drift where Pacific white-sided had historically been seen. This might be an indication of a shift in distribution known as a range shift. Using remotely sensed satellite imagery, we can find sea surface temperature at each location where Pacific white-sided dolphins were acoustically detected in drift 7 and 8, and observe that the temperature of Pacific white-sided fell commonly between 13 to 14 degrees Celsius. Using the data found on the prior map, the box plot shown on the left with Pacific white-sided dolphin location on the x-axis and the average surface temperature on the y-axis showed that the location of Pacific white-sided dolphins in 2018 were found in a narrow temperature range between 13 to 14 degrees Celsius. The GIF on the right shows monthly temperatures during August, September, October, and November in 2018 during the survey time period, with preferred Pacific white-sided temperatures 13 to 14 degrees shown in blue on the map. Preferred temperatures were always located north of Point Conception, which mirrors the results of acoustic data giving evidence for a northern range shift for Pacific white-sided as a response to a warming climate. Pacific white-sided are found less often in the south, which may be related to a rising ocean temperature. Greenhouse gases trap excess heat in the atmosphere, which is then absorbed by the ocean. 90% of excess heat created by greenhouse gases are absorbed by oceans. California oceans alone have increased at a rate of 0.2 degrees Fahrenheit per decade. La Jolla has experienced an increase of 0.6 degrees Fahrenheit per decade since 1973. One of the many results of rising ocean temperatures is a range shift for marine mammals. On the right shows a plot of a theoretical range map shown in green of cetaceans that prefer temperate habitats, with white showing regions that are uninhabitable and too hot. The left side of the data of the map shows data from 2001, and the right side shows the habit shift that would occur if the temperature of the ocean increased by 5 degrees Celsius. This demonstrates how range shifts have happened and are happening on a global scale. Understanding the range shift of Pacific white-sided and rhesus dolphins will be a useful prediction to understanding the impacts of climate change on cetaceans in the California current ecosystem, with minimal monitoring as our climate progressively increases its temperature. Climate change is driving shifts in marine ecosystems, which calls for shifting marine protected areas and fishing areas that are no longer relevant with these range shifts. We must understand how marine mammals of the past have shifted location in order to protect future marine mammals in our warming climate. Thank you so much. And if you have any questions that I can't answer now, I have my contact information there too.
We have time for one question, either online or. There aren't any questions, let's move on. Thanks, Alex.
Thank you guys so much.
I'm gonna use this tool today just to make it easier. Oh, we have a question. Are there Pacific marine, sorry, I lost it.
Marine protected area.
Yeah, that are you referring to?
I'm mainly referring to the ones that in the areas that I researched along the California coast, but specifically, I am not sure. I just was talking more in like a future range. If using this data to help kind of shape future, climate protection, that's where we should look to. Thank you. Sorry.
Before we get started presenting the rest of the projects, I would like to introduce the instrument that is included in all of our research, which are called HARPs, or high frequency acoustic recording packages. At the top, the flotation keeps the hydrophone about 10 meters above the seafloor. And the hydrophone can be considered as the ears because the hydrophone collects sounds and transfers those sounds or recorded data to the data logger. And the data logger is the brain. And the data logger transfers the data onto a hard drive. And I will be sharing what an LTSA is. Okay, sorry, I'll move it later. Throughout our research, we're going to be showing LTSAs and spectrograms, and an LTSA is a long-term spectral average. And on the x-axis, it shows the time in hours, and the y-axis, it illustrates the frequency in hertz, and it indicates the intensity in colors. So if I were to get a closer look at an acoustic call, I would go into this area right here. And just to look at a deeper area, I would press that section and look into the spectrogram. And in the spectrogram, it allows us to get a closer look and understand and visualize what sounds actually look like. The spectrogram, you can listen to the sound within this seconds, or on the x-axis, it shows the time in seconds, and the same y-axis as an LTSA. And when the HARP is retrieved by the scientists, at Scripps, a form of data is sent to the scientists in X-Waves and softwares like Triton, developed by Dr. Sean Wiggins, made this program just for scientists to visualize sounds. I've been in SeaTech for 3 years now, and I am a COVID child. We did not have the chance to come down 2 years ago, my sophomore year, to present to everyone our research. And I was not able to take this class this year, but SeaTech gave me the opportunity to come down and finally give my presentation in person. So I'll be presenting a short summary of my research that was based off of the effects of environmental factors on odontocetes vocalizations in the Gulf of Alaska 2014 and 2019. This project was just based off of the sea surface temperature increase and how that impacted the phytoplankton. And with the increase of phytoplankton, it affects the whole ecosystem. And I mostly focused on killer whales and Barrett's beaked whales. Throughout this project, I was trying to figure out if there was a relationship between the blob and the sound production among the two species. For those of you that do not know what the blob is, the blob is it's just the warming of temperature waters in the Gulf of Alaska. And you can see it right here and how it increases throughout the months in 2019. And I chose 2014 and 2019 because 2014 was a colder year and 2019 was historically recorded as one of the hotter years in Alaska. And the HARP was located at site CB, right here, and CB is right on the continental slope, which contains a lot of marine mammal occurrences and ship occurrences as well. And this project was focused on March, April, May of 2014 and 19. Here is one of the results of the project. We have the presence of Barrett's beaked whales and killer whales. On this left side is the cold year, and on the right side is the warm year. On the top, we have the Barrett's beaked whales, and on the bottom, we have orcas. On the x-axis in each of the plots indicate the local time that is recorded, and the y-axis has the date recorded. And as you can see, in 2014, for both species, we have very few encounters. And in 2019, we have, or the presence of both species increased dramatically. And you can tell by looking at the black marks. And again, this is a very short summary. So this is my conclusion. Overall, there were a higher presence of Barrett's beaked whales and killer whales in 2019. So my original question was, is there a relationship between sea surface temperature and the sound production among the two species? And that answer is yes. If you would like to watch and learn more about this research, you can take a picture of the QR code right there, and it'll bring you to last year's recording. Thank you. Are there any questions?
Any questions for Tiana? Or one question?
So my project was on the acoustic detections of delphinids off of Southern California. And this HARP data was from 2014. Okay, this is the global location of the HARP sites video. Okay. Okay, so the HARP site is shown as the red dot on the picture. The depth of the instrument was 580 meters below sea level. On this site, they get quite a bit of anthropogenic noise from passing boats. The duration of this information was from January 6th to April 8th of 2014. What I decided to study for this project is dolphins' acoustic presence. So at the beginning of this study, I knew I wanted to look more into the Rizzo dolphins. And a little bit about the Rizzos is that their length, they can grow to be 8.5 feet to 13 feet. And the range map is shown in the top right corner. And they mostly like to stay close to shores. Their diet consists of squid, crustaceans, and other invertebrates, but they mainly like to eat squid. As the project progressed, I noticed that Rizzos do not show up as much as I would have hoped. The way I found this out was from looking for the clicks that had banding, which Alex talked about a little bit. So the picture is showing the picture in the bottom right is showing the banding. I was only able to identify a couple of the clicks as Rizzos, and I decided to keep the data, but also go through the data again and look for whistles and clicks. So we couldn't find many Rizzos. So here are the other odontocetes in the area. There's the Pacific white-sided dolphin, the common bottlenose dolphin, the short-beaked common dolphin, and the long-beaked common dolphin. So these are all the main species, and they're the pictures beside them are the range maps. How I decided on these species is based on their range maps and commonality of the species. And now a little bit more about these species is that the species lengths vary from 5 feet to 13 feet. The smallest being the short-beaked common dolphin that can grow 5 feet to 9 feet, and the largest being the common bottlenose dolphin that can grow up to 13 feet. They all have a similar diet consisting of shrimp, squid, and small groups of fish, schools of fish. And now how they sound. They make a variety of sounds, echolocation clicks, which they use for navigating and foraging, buzzes and whistles, which they use for social calls, and a different type of whistle, which is the signature whistle, which I will talk about later. And now I will play a sound of whistles and buzzes of the short-beaked common dolphin. Okay, so what the heck is a signature whistle? So going through the data, looking for clicks, I came across whistles that were repetitive, and it made me wonder if they were signature whistles. So what the heck is a signature whistle? What does it mean? The way it is identified is by its distinctiveness and repetitive whistle pattern. And on the screen shows some examples. And where I got this from was from a paper that was made by Layla Saint. So these are usually used for social purposes and individual dolphins to recognize each other. Okay. So my goals for this project was find what the click pattern is like during the day and night and explore odontocete whistles and look for patterns. And my methods. I started off by going through the first round of LTSA scanning and finding click encounters and logging them in Triton. In total, I logged 329 click encounters. And then I went through for a second round of scanning to look for whistling encounters, and I logged 217 whistling encounters. Okay. So this is a graph of click encounters. The x-axis is showing the hours of the day, and the y-axis is showing January through April. The shaded area is showing nighttime, and the bright area is showing the daytime. So this is showing that the clicks usually showed up at night, which is to be expected because dolphins usually look for food at night. Okay. So this is a graph of whistle encounters. That they may be signature whistles, but we are unsure, so we are going to be calling them stereotype whistles. This has the same format as the other graph. The x-axis is hours of the day, the y-axis is January through February. It's shorter because we weren't able to go through the rest of the data. And the solid red line is showing the whistles, which are just normal whistles. And then the dotted lines is showing stereotype whistles. And the 1, 2, and 3 show that there is like more than one stereotype whistle. The 2 stands for 2 stereotype whistles shown in that encounter. Okay. So these are some examples of stereotyped whistles. This top left photo right here is of an LTSA, and this is just showing where I clicked to get into the whistles. And I'm gonna play the sounds of each. Okay, and I'm going to be playing the second sound. And now for the last. Okay. So my first study goal was find what the click pattern is like during the day and night. So the data showed that the echolocation clicks mostly showed up at night. And this is important because it shows that if this habit changed, we might wonder what might be changing or happening to make this occur. For example, if we started noticing that the echolocation clicks started showing up during the day, we would wonder if their food sources were changing. And my second study goal was explore odontocete whistles and look for patterns. We found that these stereotyped whistles would usually show up at night, similar to the echolocation clicks. What we didn't find were Rizzos. And this is interesting because it leaves us with tons of questions. Like, why aren't they showing up as much? Does it have to do with food sources, environmental sources? And some of my future projects that I'm looking into is going more into depth and looking more clearly at characteristics of each whistle and characterize them. And my second option would be, is there a correlation between clicks and signature whistles? Thank you, and do you guys have any questions? What is a stereotype whistle? Sorry, I'm choking. Okay, so what is a stereotype whistle? A stereotype whistle is basically a signature whistle.
where there's like a pattern or something like that. So.
Sorry. Good afternoon. My presentation was on acoustic analysis of blue whales and fin whales in the Santa Barbara Channel in 2014. I had the same site area as Keisha, but I listened at different frequencies and 2 different species. And my first species was Balaenoptera musculus, also known as blue whale. It's the largest animal ever to live on Earth, making it the largest whale. It can grow up to 100 feet and weigh about 290,000 pounds to 330,000 pounds. They call at around 50 hertz, which is a really low frequency that you can't normally hear. So when you're listening, you have to speed it up to hear it. They are near Southern California from May to September. And this picture here is a picture of the range. It's a range map of where in the world they live, which is pretty much everywhere except for cold and icy waters. Here is an LTSA and spectrogram of blue whale encounter and calls. On the x-axis of the LTSA, the length time is 1 hour, and on the spectrogram here, the time is in seconds. It's about 60 seconds. Here is where I would click to get into the spectrogram, and also where I would start an encounter. And here is how they sound. My next species was Balaenoptera physalis, which is also fin whale. It is the 2nd largest whale species next to blue whale, and it can grow up to 85 feet and weigh up to 160,000 pounds. They call at 20 or 40 hertz, which also when you're listening has to be sped up to hear it. And they are in Southern California year round, but more common in summer and fall months. And the range map is exactly the same as blue whales. Here's an LTSA and spectrogram of what the calls would look like. And it's the x-axis for both is the same as a blue whale. And here the square is where I would click to get into the spectrogram to listen to the call. And here's how they sound. So I chose blue whales and fin whales because when Mr. Mahoney was having us just learn how to use MATLAB and Triton and how to log calls, I found 2 different calls at 2 different hertz, but it was at the same time. And so I was interested in what they were, and Mr. Mahoney had thought they were blue whales and fin whales. So when it got to choosing what our project was about, I wanted to look at when they're in an area at the same time. So that is the goal number 1, which is overlap of blue whale and fin whale encounters. Goal number 2 is duration of encounters, and goal 3 is time of encounters. My methods, I use MATLAB and Triton, and I use the logger method in Triton, which is here, and I would log the start and end time of an encounter. And to log an encounter, I would start at the first call and end at the last call. Then if there was a space more than 30 minutes elapsed, then I would start a new encounter. And I logged about 170 encounters in total. This graph is the percentage of overlap calls and non-overlapping, which you see on the y-axis, and the percentage in total of the encounters, which is the, wait, sorry, I messed up. Non-overlapping and overlapping is on the x-axis, and percentage of encounters is on the y-axis. And here in the blue, here and here is blue whales, and the orange and or red is fin whales here and here. And as you can see, there is more blue whale encounters compared to fin whale. And because of that, the overlapping took more time of fin whale encounters that I found. So that's why there's a bigger percentage for overlapping. This shows the number of encounters, but doesn't tell us how to log the encounters. So that leads to this, which is the encounter call length. You can see here on the top is blue whales and fin whales, and on the x-axis, it's how long each encounter was in hours. And then the y-axis is the number of encounters that were however many hours long. So we stack them up here to show how many times an encounter was the amount of time. So here's 1 hour. So this is how many calls are 1 hour long. And it's the same for fin whales. And we noticed that there is a lot more fin whale encounters compared to, there's a lot more blue whale encounters compared to fin whales. And we also noticed that after the 2 hour mark, which is here where the arrows are pointing to, there's only 4 calls that are longer than 2 hours for blue whales. And there is 12 calls longer than 2 hours for fin whales or encounters. So we looked at overlap, which led to look at encounter duration, which now leads to the time of encounters. Here you can see that the gray right here is nighttime, and in the middle is daytime, which is shown on the hours of is shown on the x-axis, which is hours of the day, and the y-axis is in months. And you can see that they were calling at very different times in months, and it didn't really matter when they were calling throughout the day. And we had also noticed that when there was blue whale and fin whales together, which is shown here, the fin whales weren't as calling as long as they were down here. So it's interesting to look at. And the gray space in the middle is shown that there was no data collected and no recording during this data set. To summarize, the result from goal 1, which is overlap of encounters, showed that there was very little overlap, but there was more time of overlap taking in the fin whale encounters. And the result from goal 2 was that there was very different numbers of encounters, and blue whales had a lot more, and the durations were pretty different, and most for fin whale were longer. And the result from goal 3, which was time of encounters, was it shows that there was very different times that they were calling and more busy. And we also saw that there was a lot of time missing that I'll be able to look into for a future project and find a different deployment set that has the time that I'm missing to find out if there is overlap or no overlap in that time. And we decided that the importance of this project was that they could be fighting for prey, and the overlap could mean something, and we hopefully can look into that more next year when I continue SeaTech. Guyana, is there any questions? Yes.
So I find it surprising that you it sounds like you either hear blue whales or you hear fin whales, but you don't often hear blue whales and fin whales.
Yeah, that was most of it. I went through like every single disc in our deployment set, and I rarely found them together.
So we're sort of like conditioned to think that we hear these animals from so far away that you would expect that you hear whales and whales. So my question is, when you often find that there were very faint sounds maybe of one or the others, or just like nothing?
Yeah, it was pretty much nothing. When there was a blue whale calling, you couldn't really see any sound from anything else besides like anthropogenic sound.
That's a really interesting result. Great work.
Thank you.
Does this one work?
Yeah.
Okay. Hi, this is. Hi, this is our project about the characteristics of narwhal whistles in the Eastern Canadian Arctic by me and Haley. So this video shows our area of study, which is in the Northern Canadian Arctic in the Nunavut region on Inuit Indigenous land.
So in the area of study, we're looking at this body of water, and we were looking at 2 hydrophones to get our data. And so the first hydrophone is MI, and that stands for Milne Inlet. And then the second hydrophone is PI, short for Pond Inlet, because it's near the community of Pond Inlet. And the third point on the map shows Mary River Mine, which does a lot of shipping throughout this body of water. And that's a possible concern for the narwhals because it may mask their social communication.
This is a narwhal. It's an odontocetes. There's only 2 species in its family of Monodon montidae, and it's the narwhal and beluga. They have a grayish body that gets darker as they age, and they have a tooth or a tusk that is on their head. Males mainly have this, but some females have it as well. They have 2 main predators, which are humans and killer whales, and they are important to the Inuit for food, social, and ceremonial purposes.
And so this map is the range map for the narwhal, and so that's what the yellow highlighted region is. And the black circle shows where we're looking at. And that's an important summering ground for the narwhal. And because the rest of the year it's covered in ice. So during winter, they move more south.
This is the behavior of narwhals. Their main diet consists of fish and squid. They're capable of diving up to 1,000 meters in depth. They use echolocation clicks for foraging and navigation, and they use lower frequency sounds such as whistles for their social communication. Narwhals are vulnerable to different threats, including climate change and anthropogenic noise. And this is what they sound like. Okay, we had 3 main objectives. The first one was what is the average start and end frequency of the whistles? And are there any differences between the start and end frequency between both of our sites? And do narwhal whistles and ship noise overlap in their frequencies?
So the importance for our research was because there's a lack of information on 2 things, on the lower frequency narwhal whistles and how ship noise may possibly affect that. And so when ship noise can possibly affect that, their social communication is masked, and that creates many issues.
We used Triton, which is a program on MATLAB, and we collected our data from 2 sites, as Haley was showing, from Milne Inlet and Pond Inlet as well. We used clicking events to identify the areas where narwhals were present, and we used a data logger to log the whistles and their characteristics. We used 4 different deployments, and as you can see, the start date and the end date were pretty similar in all of the disks. More of our methods. We had a 2-hour plot link for the LTSA, and we went up to 40 kilohertz as well. You can see the black box that shows the clicking events of the narwhals. And for the spectrogram, we normally had 30 seconds while looking through the data. But once we found a whistle, we shortened it down to 5 seconds to increase the efficiency of logging the whistle. And how we log the whistle, we click the start of the whistle and log the start hertz and also the start of the time. And we do the same thing for the end of the whistle and log the end of hertz and also the end of the time. And we save every image and every audio and log it.
Sorry. So from our results from the PI site, we logged 418 calls, and the top line on the graph shows the maximum frequency recorded, and then this bottom line represents the minimum frequency recorded. And this is without the outliers, which are the dots on the top. And the black line in both of these shows the median, and the mean or average start frequency was about 1,900. And the end frequency was 1,750. And the x-axis shows the start and end frequency, and the y-axis shows the frequency in hertz. And so from the beginning to the end, the frequency in the beginning was slightly higher than the end, which is what we saw when we were logging this data.
For Mellon Inlet, we had a total of 455 calls altogether. And the grayish box is the start frequency and the brownish box is the end frequency. The frequency goes up to 5 kilohertz, and the black bold line is the median for both of the boxes. And the average was about 1,886. And for the end frequency, the average is about 1,779.
And so with the two sites, like the total of the two sites, there were 873 calls logged, and it's basically the same graph with the maximum, minimum, and median, and then the outliers. And so the start frequency in total was 1,893, and the end frequency was 1,765. And the data still showed that the start frequency is higher than the end frequency, which was similar to what we saw when we were looking through the LTSAs and spectrograms. And we used a Wilcoxon test, which is a statistical test, to compare or look at this to see if our data was similar or different in the start frequencies and end frequencies. And so in the start frequencies from both sites, they were similar, and same with the end frequencies, but the start and end frequencies were different.
For our conclusion, we gathered the information. We estimated the average start and end frequencies of the whistles in both Milne Inlet and Pond Inlet. And we also found out that there is no significant differences of the whistles from the start and end frequency between both sites.
And so we also saw that shift noise may mask the normal communication whistles. And so that's what this LTSA shows. And we were looking at it in 6 hours on the y-axis and 40 kilohertz on the y-axis. And the line right here shows the maximum frequency for the whistles, and then the minimum frequency for the whistles. And the shift noise, the shift noise may mask the normal whistles because they're within the same frequency bandwidth. And yeah.
We have two main future study questions, and the first one is to undertake more research to better understand if anthropogenic noise is able to affect the normal whistles, to affect normal social sounds such as their whistles and their echolocation clicks. And the second question is to further research to better describe the characteristics of their whistles, such as the duration. Are there any questions?
Yeah.
Because first of all, this is really amazing work. So I work in this area on this particular project, and I'm really excited to see you guys do this work. One question, this is do you know which type of vessel that was? Is that an icebreaker? Is that a coal carrier? And did you look at different kinds of vessels or just one vessel?
Can you go back to the
Yeah.
LTSC?
This one. We used a random picture, so. Oh, sorry. We're not sure, but it'd be cool to research about that as well.
That is the commercial assistance icebreaker bot. And so the 26th of July, there's a teeny little bit of ice left in the with the bot. It looks like it's the Bosca, the icebreaker, and maybe
That's the big one. Great.
Oh yeah.
Before you leave this
Yeah.
plot, there's one thing, many things I think are really interesting about the project. And thank you for answering the question. Does ship noise overlap with the social sounds of narwhals? There'll be people whose waters these are and whose wildlife this is. We'd be very interested and happy to hear finally a definitive answer to that question. Which is ships stomp on narwhals. At least it's there's another thing that I actually haven't seen with my own eyes until I looked at this plot. But is that to me that looks like echolocation, narwhal echolocation in the top left part of the plot. And to my eyes, I also see that the ship noise appears to go up into and overlap with the narwhal echolocation. Is that something that you saw repeatedly, or just the only instance, or did you notice that?
When I logged this data, I only saw this happen twice. And the first time I saw it, like the echolocation clicks were still going while the ship was passing. But in this picture, it shows that they stop when the anthropogenic noise is coming towards them.
Yeah. But that was out of our research.
Hello? It's not on. Great. Hello? Hello? Okay. God dang it. All right. Okay. Hello. My project is basically to determine whether anthropogenic or human-made noise has any effect on sperm whale presence in the Western Atlantic. However, if you don't know much about sperm whales, here's some basic information. Their scientific name is Physeter macrocephalus. They're the largest toothed whales that we know of, and their prey primarily consists of squid, although they also eat octopi and deep sea fish such as sablefish. The average adult male weighs about 90,000 pounds and has a length of 60 feet and a lifespan ranging from 60 to 70 years. And if you want to know what they sound like, simply click your tongue and amplify that sound by 20. You've got it. So sperm whales live basically anywhere in the world except for the most extreme polar regions, but we're studying them in a specific region in the southwestern Atlantic. Where, according to Stan Street et al. 2018, sperm whale presence is higher during the winter when the ocean is cooler. The two sites we had for our study were GS, which stands for the Gulf Stream site, which is off the coast of the Carolinas. The Gulf Stream is important to study because it cycles phytoplankton and warmer water throughout the Atlantic Ocean. And the more phytoplankton there is in the ecosystem, the more productive that ecosystem tends to be. At GS, there was a 12 kilohertz echo sounder occurring during the first 3 to 4 months of every recording year, which, if you don't know, an echo sounder is used for a variety of purposes and is produced by a transducer in either the hull of a ship or a part of a moored instrument. And since there wasn't any consistent shift in the data, we assume that it's from a moored instrument. And at the same time the echo sounder was active, there also was a decrease in sperm whale presence. And the other site we studied, which was BP, was our control site. And we chose it as our control site because of its close proximity to GS, as well as its similar depth and lack of the 12 kilohertz echo sounder. Because the echo sounder was within the same frequency range as the sperm whales, and this decrease occurred for the sperm whale presence at the same time as the echo sounder was active, we made us wonder whether the echo sounder caused the decrease. So to determine this, that's what we decided to study, whether there was a difference in sperm whale presence when the echo sounder was active compared to when it was absent. However, we also decided that environmental factors such as sea surface temperature or chlorophyll A concentration could have played a role in their decrease. So we studied that at GS during the same time as well. Here's some general info about the HARP deployments. Each site had 3 deployments ranging in depth from about 930 meters to 1,005 meters. And they started continuously recording from April of 2016 to June of 2019, resulting in 1,100 days total of continuous recording for each site. However, each deployment was too big to look at all at once, so each deployment was split into 6 different segments so I could log the echo sounder. And these are 2 examples of what I might see when logging. The top 2 images are LTSAs, and the bottom 2 images are spectrograms zoomed in at the points shown. And then right here, you can see the broadband impulsive signal of the echo sounder, and then all of this is sperm whales clicking. When logging the echo sounder, I manually logged in Trident, and I looked in the frequency band of 5 kilohertz to 15 kilohertz. And some things I looked for were that the peak frequency was at about 12 kilohertz and that the echo sounder occurred at a constant interval of every few minutes or so. And also another thing to note was that the echo sounder was also a lot more isolated than sperm whale clicks, with sperm whale clicks occurring in intervals of 45 minutes, which is the typical length of their dives. Unlike the echo sounder, we use an automatic detector for sperm whale clicks, and then we use a program called Dead Edit to clean the detections up, to basically ensure that we were only using sperm whale clicks for our study and not taking into account any other odontocete or anthropogenic noise. So here is an example showing the average weekly sperm whale presence and the average weekly echo sounder duration at the Gulfstream site, with the bars being the sperm whale presence from 0 to 600 minutes and the red dots being the echo sounder duration from 0 to 150 minutes. Although this echo sounder duration really ranged from 0 to 85 minutes, with one outlier right here in March or April of 2018. And then this also right here, you can see the noticeable gap in 2018 in sperm whale presence at the same time as echo sounder, which sparked this study. This plot is the same type as the last plot, except it's for the control site, the Blake Plateau site, and there's the noticeable lack of echo sounder in the data. It's also important to note that the y-axis only goes up to 150 minutes, indicating that there is overall less sperm whale presence at BP compared to GS. After making these observations, it was time to do the statistical analysis, which to do, first you had to make a null and alternate hypothesis, which was the same for both comparing between the 2 sites, with GS being times with echo sounder and BP being times without at the same time, and also comparing within just GS, comparing different times with and without the echo sounder. The null hypothesis would be that there was no difference in sperm whale presence, and then the alternate hypothesis would be that there was a difference for times with echo sounder and times without. To determine if there was a statistically significant difference, we used the Kruskal-Wallis test, which is typically used to determine if a difference is statistically significant between the medians of 2 or more independent groups. And then once you run that test, if you get a p-value of less than 0.05, then you could reject the null hypothesis and consider the difference statistically significant. Once you ran the Kruskal-Wallis test, we put the results into a plot as shown. This one shows the results of comparing between the 2 sites, with GS times with echo sounder on the left and BP times without the echo sounder on the right, and the y-axis being normalized daily sperm whale presence, which basically means that the more daily sperm whale presence there was, the higher the values. Looking at the 2 box plots, they overlap a lot, which is reflected in their p-value of 0.0675, which is greater than 0.05. Thus, we accept our null hypothesis and the difference is not statistically significant. This other plot is comparing within GS itself with echo sounder on the left and times without echo sounder on the right. And again, the p-value is greater than 0.05. Thus, the difference is not statistically significant. However, If that decrease in spermal presence is not due to the echo sounder, then what could it have been a result of? That's where the environmental data comes in. We got sea surface temperature from the multi-scale ultra-high resolution satellite, and we got the chlorophyll A concentration data from the NOAA VERSE satellite. And after getting all of these different variables, we decided to combine them all into one plot during our time period of study at GS. And here it is. It's a lot to look at, I know. So I'll just walk you through it. The black bars are the average weekly spermal presence from 0 to 600 minutes. The red dots are the average weekly echo sounder duration from 0 to 150 minutes. The blue line is the sea surface temperature from 22 to 31 degrees Celsius. And the green line is the chlorophyll A concentration in milligrams per meter cubed from 0.05 to 0.5. Looking at this plot, it appears that the sperm whale presence is closely associated with both environmental variables, with presence increasing during the warmer sea surface temperatures and higher chlorophyll A concentrations. And it's also important to note for possible future discussion that the major sperm whale presence decrease in 2018 occurred during the coldest portion of the 3 recording years. So after all that, that's a lot to take in. So let's just do a quick summary of the results. We looked at 2 sites, the Gulfstream site and the Blake Plateau site. And overall, we found that sperm whale presence was much higher at the GS site compared to BP. And at GS, the 12 kilohertz echo sounder was present during the first 3 to 4 months of each recording year. And when we ran a Kruskal-Wallis test to determine if there was a statistically significant difference, we determined that the difference was not statistically significant between times with echo sounder and no echo sounder. But we did find that sperm whale presence seemed to be closely tied with sea surface temperature and chlorophyll A concentrations. So what could possibly explain our results? Well, one explanation is that sperm whales don't care about the echo sounder at all, because there is anecdotal evidence to suggest that sperm whales are unaffected by anthropogenic noise, and even though this echo sounder was directly within their frequency range, they could remain unaffected. Another possibility is that BP wasn't a good control site with much less sperm whale presence than GS, and instead of picking one of the southern sites, we should have picked one of the northern sites instead. The third possibility is that sperm whales could have a delayed response to the echo sounder. So by comparing times directly after the echo sounder was active, we could be convoluting the pattern and skewing the results. And the final possibility is that the low sperm whale presence was due to environmental factors. So like the sea surface temperature or chlorophyll A concentration, which leads me to another reason why this study was important. Because if sperm whale presence is really heavily affected by sea surface temperature, then the change in climate could lead to changing sperm whale behavior and affect the ecosystems they play a role in. So we need to be asking questions in the future, possibly about whether we need to change fishing regulations of their prey or changing their possible exposure to harmful fishing methods such as drift netting, or just changing fishing grounds in general. So with all that said, what could we do in the future related to this project? Well, there are other more robust statistical methods that we could have used given more time that predict how variables affect sperm whale presence. Or a question we could ask in the future is whether the environmental variables such as sea surface temperature or chlorophyll A concentration truly affect sperm whale presence, or other environmental variables, especially focusing on ones that reflect the deep sea environment, since that is where sperm whales spend most of their time. And a final question we could ask in the future is whether a sperm whale's age or sex affects how they react to variables, especially anthropogenic noise. Thank you, and are there any questions? Okay.
So in those class, it looks like every point of the 12 kilohertz things just disappear. You know about how many things or about how long it took for it to disappear?
You mean the fact that like after, how many times it activated?
So there's what the experiment is. It's a release when the acoustic release is at the warning. It says somebody left it on. You got before it's about
I don't know that. I'm sorry. I wasn't keeping track of how many times it activated, but yeah, I don't know. All right. Tell me in the future. Thank you. Huh?
Would you share your logs with him? Could you send your logs to her?
Yeah.
Do you think it's so hard?
So what happens if you don't disable release the professional data? It will clean the water if something makes the right sort of variation there, something that can be variation. They should disable after 5,000 things, but I'm curious if you're getting the regular thing. Sometimes we actually seen it in Arctic data sets with belugas. We'll see it with dolphins where they'll approach activated release and they'll, when it makes a sound with them, it makes a sound with it.
Yeah, we actually didn't know what the source of the echo sounder is, so if that's the source, that would be pretty funny. Honestly. So, all right, thank you.
So I'm Jordan David, and my study focused on the effect of ship noise on humpback whale behavior in Southern California. So my study surrounded the humpback whale, or Megaptera novingulae, and as you can see from the picture on the left, humpback whales can be found in all oceans except for the polar seas. They prefer shallow waters, and when they want to mate, they go to several locations around the world with warmer waters. And they are rorquals, as they eat by lunge feeding, and their main food source is krill. They can grow to be 50 feet long and around 55,000 to 66,000 pounds. And their name comes from the hump on their back that forms when they begin to arch their body to begin feeding. So some background on the humpback whale. Humpback whales evolved around 45 million years ago, and in 1931, the first container ship, the Otto carrier, was launched. So that means that humpbacks were around container ships for 91 years, or a total of 0.000002% of their entire existence. So right now, there are around 5,400 container ships in use in the world's oceans, and around 90,000 ships make up the commercial fleet. So this picture shows the amount of commercial ships that are currently in the water as of April 2022. The green arrows represent cargo ships, the red tanker ships, the blue passenger vessels, and the rest of the arrows and colors are divided up into several other types of ships. So my study occurred in Southern California, shown in this picture to the right. And this section right here is a zoomed out image of the exact area of the state that my study was located in. And what makes this area interesting is that a national marine sanctuary is right next to the busiest shipping lane in the Western Hemisphere. So Site B, this star right here, is the location of my HARP, and it was at a depth of 580 meters on the northbound shipping lane. And my study was conducted from October 2013 to January 2014. So the overarching research aim of this study was to see if shipping traffic affected humpback whales. And I had three main questions during my investigation. And I first looked into the seasonality of humpback whales in this region. with my null hypothesis being that there's no difference in humpback whale presence between seasons, and my alternate hypothesis being that their humpbacks are present in the spring and fall, which is based on previous work by Tyler Helby. My second question had to do with overlapping of shipping with humpback encounters, with my null hypothesis being that the encounters would be around 50% in the presence and 50% not in the presence of ships. And my alternate hypothesis being that encounters would be about 16% overlapping with the ships, which is just my guess. And my third question has to do with the acoustic behavior of humpback whales and the presence of ships, which is, does the humpback whale behavior change in the presence of ships? With the null hypothesis being that there would be no change in the behavior of the humpback whales in the presence of ships, and the alternate hypothesis being that the humpback whales would simply stop calling in the presence of ships. So while humpback whales are well known for their complex songs specifically, they make a variety of different sounds. They make both song and non-song related calls, and there are several types of non-song related calls that are classified. These calls are moans, grunts, pulse trains, blowhole associated sounds, and song. Only male humpback whales sing, as far as we know of, and while because of this, it may seem like the song serves some type of mating purpose, it is assumed that it serves a variety of different roles. Their songs can be around 80 to 4,000 hertz, and these are some examples of sounds that are in the humpback whale repertoire. So Tiana already explained what a spectrogram, Chiden, and LTSAs are, so I will go into detail with what exactly we did with this data. I logged both humpback whale encounters and ship presence, and I used a system of analyzing the data to determine the relationship between the humpback whale calls and ship noises, which were before, during, and after the ship. I did this to determine the relationship between the humpback behavior and ship noises, and to find the correlation between them and to find out why the animals are calling. This is an example of a humpback encounter. Let me play the sound. I used the same methods to determine an encounter as the rest of the interns. And here is an example of a ship. You can see the characteristic U shape on the LTSA, which is shown by this little pink bar. So just to give some insight into what I mean by BDA, before, during, and after, this is an illustration demonstrating the relationship between the calls of the humpback whale in relation to the ship noises. The blue bar represents the humpback whale, shown by this little humpback cartoon, as it extends past the ship. The red line differentiates when the ship is making noise from the calls of the humpback whale. This section right here shows the humpback whale calling before the ship. This section right here shows the humpback whale calling during the ship. And this section right here shows the humpback whale calling after the ship. So let's see how these behavioral changes look like in our data. This is behavioral type B. This red section right here is the ship. And these sections right here leading up to the ship are the humpback calls, shown in the pink circles. And as you can see, the humpback whales call before the ship, and they stop calling before the ship comes. We looked into this behavioral type because we wanted to see if the ship is potentially what made this whale stop calling. This is behavioral type D. This section right here is the ship, and you can see the humpback calling during the ship. And, as you can see, the humpback continues to call during the ship noise. We looked into this pattern to see if the ship sounds had no effect on the humpback whales. This is behavioral type A. This section right here is the ship. And as you can see, the humpback calls occurred after the ship. We looked into this ship type to see for the possibility that the ships made the whales start calling and to see if maybe the ship sounds made them stop calling and if they just like started calling again after. So this plot shows the humpback calling presence during the fall and winter of 2013. During this time, the HARP was recording continuously throughout this data range. The x-axis represents the dates, and the y-axis represents the calling presence of the whales in hours per day. The blue bars represent humpback calls, and a lack of bars represents a lack of calls for that day. The maximum number of calling hours per day was 23 hours, and the minimum was 0. And as you can see, the majority of the humpback calls occurred during October and decreased into the month of November, with December hardly recording any calls. This means that we accept our null hypothesis because there was seasonality to these humpback calls. This graph shows the number of ships during humpback encounters, with the salmon color being 0, the green section being 1, the blue section being 2, and the purple section being 3 ships in an encounter. So as you can see, less than 5% of encounters had 3 ships. Around 18.75% of encounters had 2 ships. Around 38.75% of encounters had 1 ship, and 37.5% of encounters had 0 ships. Overall, over 60% of humpback encounters had at least 1 ship. So this plot shows the total humpback whale behavioral calling changes caused by ship noise. The x-axis represents the different calling changes before, during, and after, and the y-axis is the total count of the changes that I found in our data in that period of time. The green bar for before a ship is the smallest, with a total count of 22. The blue bar for during a ship is the second largest with a total count of 25, and the salmon bar for after a ship is the largest with a total count of 28. And even though the bars are at different numbers, there's no one bar that dominates the rest, which was surprising to us as we thought that there would be one response to ships, whether before, during, or after, but it looks to be spread about equally. So now for my conclusion. I found that most humpback whale calls occurred during the month of October. Humpback whales and ships are in each other's presence around 60% of the time. And lastly, I found that the presence does not have a large impact on humpback whale acoustic behavior, as there is not much of a difference between whether the whales call before, during, or after a ship. So now that we have looked into our results, let's talk about its importance. First of all, this study helped to identify the relationship between ship noise and humpback whale acoustic behavior. That is, in this study, there is now one behavior change that is most prominent when exposed to ship noise. We thought that these ships had a large impact, as they were present in 60% of the humpback encounters. But our data is not different enough to suggest that the ships play that large of a role in their acoustic behavior. Perhaps the whales adapted faster than what would be expected of them, or maybe they continue to call when they are near a ship, even if it causes them stress. Or maybe calling is absolutely necessary to the whales and they cannot spare to stop it even if a ship is present. It would be interesting to do another study that compares ship noise to humpback whale cortisol levels, as that can determine just how much distress the whales are in without the use of their calling. As we have seen in the study, that their calling may be necessary to them and cannot determine exactly how much stress their environment causes them. Future scientists should look into how ships continue to affect whale calls in different seasons and in different behavioral states, as that the number of container ships will likely only increase as time goes on. If their behavior is inflexible to modern threats, then humans need to change their behavior so they can remain thriving in their environment. And thank you. Any questions? Yes. So you're asking what time period did I look for BDA? Basically the entire period I was recording in, so October 2013 to January 2014. Then I just go through, and if I seen a humpback call, then I would see if it was either before, during, or after a ship. And if it was like long after a ship, I would record it. But if I seen any of those 3 behavioral changes, then I would log it, if that answers your question.
How many minutes would you? I think she means how many minutes do you consider before, how many minutes after the ship?
Well, I looked into the same amount as I did an encounter, so around 30 minutes, then I would deem that as before, during, and after. Right. So we do have a few acknowledgements to give out. So I will go get supplies and the rest of the SeaTech members will come up. First off, a huge thank you to our members, Anne Simonis, Natalie Postalgian, Vanessa Zobel, Ava Hidalgo-Pla, Eric Snyder, and Morgan Zeigenhorn, who have helped us every step of the way. We would also like to thank the Scripps Whale Acoustics Lab for providing us with access to the HARP data and support and analysis, especially Dr. Josh Jones and Professor Bob Rex and Professor John Hildebrand for their continued support of the SeaTech program. Thank you to NOAA for providing support and analysis. And lastly, thank you to the Oceans North for their support with the Arctic data.
Yes, thank you very much for coming, both our virtual people and our in-person folks. We really appreciate your support and we hope to see you here next year at this time.
And I'll just invite anyone who's still sticking around, if you have more questions that you didn't get to ask the students, you want to hear about their life or their plans for their future, stick around, we'll have a meet and greet outside. So thanks again for coming.